Good morning, everyone, and welcome to a new EBD seminar series. Thank you so much for joining us today, despite the change in a schedule and that the organization is a little bit different. So I really appreciate that you've made the effort. My name is Marijose Ruiz, and I'm going to be introducing our speaker today, and I'm also going to be moderating the questions. Those of you that want a certificate of attendance today, because I've already given you access through Zoom, you just need to send us an email and we'll send you the certificate. And remember, the email is seminars@ebd.CSIC.es. And if you want to make questions, you can do them either through the chat, but because we are in Zoom today, you can also just unmute your audio and turn on your camera, and you can make questions to David. So our speaker today is David Quevedo. He's a third-year PhD student with Paco Garcia, and he's going to be presenting us his PhD project, which talks about socio-sexual networks. So David, whenever you are ready, you can start.
Okay, thank you for joining me. As Maria Jose said, I am David Quevedo, and I will present my project thesis, Socio-sexual networks, sexual selection and population spatial structure under the focus of network theory. Okay. I want to say the main issues addressed by my thesis project are related to the evolution of reproductive interaction driven by sexual conflict. The sexual conflict arises because the difference of interest in reproductive terms which exist between males and females of the same species. The sexual conflict explains, among many other things, the evolution of traits that increase the reproductive success of one of the sexes at the expense of the other sex fitness, and often leads to antagonist sexual selection. There are many examples about sexual conflict. One of the most studied cases is the cost of mating in Drosophila females mediated by the sperm's byproduct of males. This conflict explains relevant aspects of the biology of reproduction and population viability. However, our understanding of its evolutionary implication is limited by the absence of knowledge related to the role of sexual conflict underlying sexual selection dynamics or the architecture of social and sexual interactions, which is called socio-sexual network for us. So, due to this absence of knowledge, I wanted to obtain a global view about our research in line compared with the rest of great areas of research in ecology. Reviewing the number of citations related to the main researching lines of this thesis, we found that they arise at the start of the 21st century, and it has been increasing till nowadays. However, the numbers are not so important like in other researching areas. So, the first thing I did when I started my thesis was to analyze the position of our researching area with the most popular areas in ecology in terms of publications. In this network, you can see the size of the nodes is based on the total number of publications in the researching area since 2000 to 2018. And the size of the links is based on the number of total publications where the two connected areas are mentioned together. As we can see, evolutionary ecology is poorly connected with the rest of areas, so it's a bit evident that more research is required in this particular area. On the top of this, if we build a subnetwork including the main topics of this thesis project, we find that they are not only poorly researched, but also poorly connected between them. This suggests a great gap of knowledge, as we mentioned above. So, the purpose of this thesis is to fill a bit of this gap, but how do we want to do it? Okay, the first thing is to know more about how networks are built between individuals when a clear scenario of sexual conflict is present. So, we want to characterize the structure of networks in a system dominated by intense sexual conflict. The next step is to determine if the architecture of socio-sexual networks is shaped by selection history in relation to the presence or absence of sexual selection and metapopulation structure manipulated through experimental evolution. Also, we want to explore within an approximation of selection experiments the role of metapopulation structure on the evolution of socio-sexual networks. And finally, it's known that the social group structure is a cornerstone of evolutionary processes based on cooperation and competition, including sexual competition. So we want to determine whether selection history has any influence on the ability of individuals to modulate their social environment, which is called social niche construction. Following, I am going to comment the methods which we are using to elucidate the questions mentioned above. First, I want to speak about our research organism, which is seed beetle, Callosobruchus maculatus. This is an ideal model system because it presents sexual conflict traits. The most relevant trait is located on the male's intromittent organ. This presents a spiny structure which damages the female reproductive tract during copulation. This damage is a way of control over the possibilities of female to copulate with other males. Also, this research organism presents other qualities which make it a good system, like a short life cycle with a duration of only 21 days between generations. Also, females can lay about 50 eggs, which means a high fecundity. These research organisms give us the possibility of working with large sample sizes. And finally, the traits of this research system and our needs of research is a combination which require very simple manipulation of the individuals. And now, continuing with the methods, I am going to mention the cornerstone of this thesis project, which is the main experiment that we perform generation after generation. This design consists in a 2x2 experiment, including sexual selection and metapopulation structure. Each treatment has 4 replicates, so we have 16 populations in total. And for our analysis, each line is a unit of replication, not the individuals implicated in the experiments. And for the metapopulations, we impose a 20% of migration rate in the structured population to recreate metapopulation condition in terms of gene flow. So I want to comment about a little bit of the nomenclature of our selection regimes, because we are going to see during this presentation this kind of names like NEP, NEM, EP, EM. Okay, so this is the meaning. And I also want to say that we maintain this experiment over 110 generations of selection under these conditions. Also, to maintain our lines, we isolate beans that have been inoculated in microdose of 1.5 milliliters in order to control the emergency of adults. During the period of adult emergency, we mark each microtube in order to know the age of the individual inside. And after this period, we place sexual mature individuals according to the selection regimes to let them copulate during 48 hours. Then we remove them. The beetles are stored in a climatic chamber with controlled levels of temperature, humidity, and light cycle of 12 hours day and 12 hours night. So now I want to comment the experiment, which are focused on the objectives I mentioned above. So the first experiment is focused to determine the architecture of socio-sexual networks. The first thing we do is identify the inter and intersexual contacts. So we build the social networks, like you can see here, and sexual networks. Then we include all the contacts building socio-sexual networks, and we obtain something like this. And fourth and last, we analyze and contrast the variables describing our networks, as I will comment in the analyzing system and contrasting hypothesis selection later on. The second experiment, testing if sexual selection and metapopulation structure shape the architecture of our networks. After building and analyzing the variables that describe the architecture of our networks, we compare them across the selection treatments described before in the main experiment section. Then we analyze which variables are shaped by sexual selection and metapopulation structure. And lastly, we contrast different starting hypotheses, as I will comment later. And the third experiment, the social niche construction experiment. As we commented before, the main objective of this experiment is to test whether the ability of individuals to modulate their social environment depends on their evolutionary history in regards to mating system and population spatial structure. So we place focal males, which come from the regime selection, main experiment, into a network constituted by males and females, which come from a stock population. Then we investigate whether network architecture depend on the origin of the focal male, that is, whether individuals coming from different selection regimes have the ability to shape their social environment differently. To get all this data, we record the individuals. So to analyze networks, we first place the individuals in this light case with the setup shown in the picture. We place 8 individuals per video recording set with a sex ratio of 1:1. The duration of the videos is 10 minutes, and we video record 2 groups of 8 individuals per line each generation, getting a total of 32 possible networks. Also, I will mention that only virgin individuals are used, which were individually isolated before adult emergency, as I commented before. First, we test the system recording individuals without any mark, but we obtain low levels of accuracy in the analysis. So we decide to paint individuals with a low invasive system as informed by our preliminary analysis. This marking system allows us to get much better accuracy levels. After video recordings, we combine visual data collection with a software that performs the individual recognition and tracking, and that gives us data on the individual's interactions. Combining this and computerized data recording, we build the structure of our data sets and get all the variables we need to describe our networks. This software gives us data that we process to analyze. This is an example of the movement behavior of individuals shown through heat maps, which is a good tool to visualize and analyze interactions. Also, we can build networks with the data given by the software and compare them across the treatments, as I commented before. When we have all the data organized, a PCA analysis is performed for all the variables that could describe our networks based on our requirements, and the original variables are ordered based on the contribution that they make to describe each network. To contrast hypotheses, we compare our network's attributes with distribution of these attributes in randomized networks. In this example, we randomized the sexual ID of vertices about 1,000 times to check the effect of being male on the strength of node across the metapopulation structure treatment. So the x-axis shows the fixed effect size of every randomized network, and the y-axis shows the frequency of the effect getting the represented size. The red outline represents in our empirical networks the effect of to be male on strength. The red line far outside of the histograms, and it suggests that the observed effect of being male is larger than expected by change. So the observed effect is not solely due to some random interactions. It's shaped by the metapopulation structure treatment. This was a simple analysis, but this powerful tool allows us to analyze networks in a deep and accurate way. Now, I want to comment the current status of this thesis and the schedule that we have and the schedule we have now. So at the beginning of this thesis, we had this schedule, well organized and with 3 possible collaboration in different countries. But some troubles found during the development of this project modified this schedule because I had many problems with the software optimization and the building of the recording box, which kept me busy the whole first year of my thesis. Also, I got problems related to the lockdown and the pandemic situation, with which delayed a lot of the undertaking activities programmed for the second year. And also, I got a difficult health situation, which kept me away from work almost 5 months. So the plans for this and the next year include experiments and processes which should have been finished during the last year. Speaking about the in-course activities, I'm now improving my knowledge about networks and learning about how to make an interpretation of their attributes. I'm also gaining experience on network visualization, analysis, and theoretical approach. I'm getting experience on network randomization, and also I'm collecting and analyzing data. Speaking about the future formation and my current formation, I assisted and I'm assisting to seminars and webinars given by the Spanish Research Council at the University of Seville. I also perform is programmed to perform short stays to improve my knowledge about social and sexual networks. I also have been in Congress, and I wish to be in future Congress and conferences related with the main issues of my thesis. I also coursing a group of courses related to the statistical approach and modeling of data related to network building. Speaking about collaborations, we received a lot of help from people who came from different educational and training programs. For example, Juan came with us to develop his final degree projects. Perry came with us from the University of Portsmouth through the Erasmus Plus traineeship program. Samuel came with us from the Lancaster University to collaborate through the Brighton Third Sector Training Program, and Bernat Garcia came from the University of Barcelona to collaborate with us through the High Intro Program. Currently, we are collaborating with 2 great researchers in the area of sexual selection and network analyzing, who are the Dr. Grant MacDonald from the Department of Ecology in the University of Veterinary Medicine of Budapest, and the Dr. Tommaso Pisari from the Department of Zoology in the University of Oxford. And that's all. Thanks for your attention. And I'm open to suggestions and questions.
Thank you very much, David, for a great talk. So I don't know if you have questions, you can either do them through the chat, or you can turn on your audio and your cameras and do the questions live. So I'm going to start while people decide. I don't know if someone wants to do any question, but I can start with one question. So when you were talking about the experiment design and the metapopulation analysis, I'm curious to know, how do you actually create that structure? And then do you have ways of following that there is actually a structure? I don't know, do you look at population genetic structure or other factors?
Well, we divide the populations. I'm going to get the. Okay, here.
Here, yeah.
We divide the population in different containers. So we create different populations, and the structure consists in the design of the storage of individuals.
So you don't do it the same generation. So you don't do it, for example, you start in one generation and then after 5 generations, you call it a structure. Do you know what I mean? It is something that you just divide. Okay.
No, we maintain this experiment during 4 or 5 years.
Okay.
Doing the same.
Mm-hmm.
Okay.
Yes. So it has been so it's been a structure for a long time.
Yes.
Okay.
Yes.
Yeah.
Hi.
Hello?
I think I can ask something. Well, thank you, David, for your nice talk. I actually always wondered what you guys were doing in the chambers, and now I finally know. So I have one question about the interaction, about the selection history, the effect of the selection history in building interaction networks. And I wonder, because you said that you put the animals into the video recording area, and you said that you would use 1-to-1 sex ratios, and also I guess that's a reduced number of individuals, so in a very small space. So I wonder how that whether that is representative of what happens to these populations in the field, like are there usually 1-to-1 sex ratios in their natural populations, and what is the scale of their interactions in the field?
Okay, so we have to take account that this is a kind of pest in storage products. So maybe you can find a great population into a massive storage, or you can find a small storage with a small population, but divided in the great side where they storage all the products. Okay, but really it's not studied the minimum number of individuals to represent a network. But we decide this number of individuals, first because logistic, second because we want 1 point of start, so because we didn't find literature about this. And okay, even it's a small network, there are some networks with more than 1,000 interactions during 10 minutes. So wow. I think if we only have 8 individuals, but we have replication, so we can analyze the same selection history through different replicates.
Nice, thank you.
Yes, I just want to say that, David put it very nicely. It's a combination of something that has to be doable in terms of logistic and what you can manage, and something that needs to be representative of what's going on. And it's always like a sex ratios are always a 1-on-1. So from that point of view, it's fine. And in terms of number of individuals, it's within the range that you can encounter in natural populations, certainly. But of course, I mean, we have the limitation. We are just focusing on a given sample size for that population. But our idea is once we start getting some more knowledge on what's going on on interactions, and David made a point that with this even limited number of individuals, the number of interactions are by the order of hundreds and thousands. So, but in the future, we'll build up upon this, and maybe we will explore the role of changing population sizes for the networks, you know. But I think it's a good starting point, and we are actually seeing some interesting results, and yeah, from here onwards, so we'll move forward. And I just want to say, David, I don't know if when you show the graphs and the methods regarding the randomizations, which is a great tool, very powerful tool. Could you clarify to everyone what you meant by strength? Because you were talking about the strength.
Oh, okay. I think this
I don't know if that was explained properly, and maybe it's something that maybe some listeners would like to know more about it.
Oh, here.
Yeah.
In this, okay. So the strength, the node strength, is the summarize of weights of links connect to the node. So in directed network, like the one we study, the in strength is the sum of inward linked weights, and the out strength is the sum of outward link weights. Okay, because we are working with weighted networks. I don't know if you know what it means.
Well,
You can explain it if you want, but basically it's just an example of one of the. I think your idea here was to give an example of for a given trait of the network that is critical for characterizing the network. So,
Yeah.
And
yeah.
the way you can analyze whether it's really significant or not using this randomization approach, which is basically setting up your null hypothesis with the real data. You've got the real structure of your data, the sample size you are dealing with, and so it's pretty cool because it's just creating your own null hypothesis to test your real data that you get from your network. So I think that's a good point. And it's just an example, but I don't know. It's up to you if you want to explain other features or not, but I think the point has been made.
Yes, for example, if we will want to know if larger males only contact with other large males and not small males, we could create a randomization based on it and recreate a scenario of social contacts based on the selection regimes. Is one of the options, but there are many options to describe the networks through this contrasting hypothesis system.
So there are two questions in the chat. I'm going to make Gemma Trigos and Miguel Fortuna. I'm going to make Miguel's question first because it's directly related to this, and then I'll ask you Gemma's question. So Miguel Fortuna is asking, it is still not clear to me. What does exactly the strength of the interactions in the networks of the links represent? It is the time a male and a female spend together?
No, it's the number of the contacts. So imagine two nodes inside a great network, and one node is really small, and the other one is really big. Okay, so the big one has a great strength because it's a node which receives a lot of interactions. Okay, and this small node receive low interactions.
But okay, he's referring to the links, not to the nodes themselves. which are different things.
The links, the
yeah.
links. Yeah, I know. The links represent a contact between two individuals. Okay.
Yeah. So then the
Yeah,
more
but
contacts two individuals make, the wider is going to be that link.
Yes. And greater will be the node.
And then greater, yeah. But the nodes depends on all the
because all
interactions that come.
yeah
And the
yeah,
links are just two of the, like how two nodes relate. So the
Yeah.
link tells you how two nodes relate, and a node represents everything that comes in.
Yeah.
Yeah. I don't know if Miguel, it is clear now. If it is not, just let us know. I'm going to, so Gemma also has a question, is not related to the networks. So nice talk and nice study, congrats. I have a question about temperature. In arthropods, the temperature regulates their activity, interactions, and also eventually influences the mating probability and success. Have you taken into account this factor, or do you plan to do it in the future? Thank you.
About the temperature when we are recording individuals.
Yeah.
Yeah, I measure the temperature inside the climatic chamber, and I measure the temperature inside the light case after turn on the lights during 15 minutes, and it's really similar. So there are no difference between the temperature inside the climatic chamber and the light case.
Mm-hmm. And do you think in the future it would be interesting to analyze the same, once you know the patterns that happen at this temperature that you are using, do you think it would be interesting in light of climate change or other things to analyze what would happen under different temperatures?
Yeah, in fact, there is a current collaboration with people from Valencia who are working with this kind of variables and who study the influence of temperature and other variables on sexual conflict. And yes, there is a program a collaboration with them.
Oh, nice. Good. Yeah, I don't know. Does anyone wants to make any other question? Oh, Miguel says that okay, that thank you for the answer. So Miguel is asking, what is the size of each network? 8 males and 8 females?
No, 4 males and 4 females.
Okay. Oh yeah, because it was 8 individuals.
Yeah, in total.
Yeah, so it is 4 males and 4 females. Okay. So if no one, do you want to say something else, David or Paco? Because I don't know if we have more questions. Okay, Miguel has another question.
Okay.
Are you thinking on aggregate some of them?
Pardon?
Are you thinking on aggregate some of them? I'm not
aggregate sure. The network. Is speaking about the network.
Because, so because 4 times 4 is a small size to make inferences.
Yeah. Yeah. Yes. We could think about to increase the number of individuals inside the recording box, but it's difficult by the logistic trouble it represents. Okay, and we also plan to record many Petri dishes in the same time. But now the factors like the software, the camera, and the space we have limit the number of individuals we can place into the Petri dish. Also, the software is limited by the number because we only can place 12 individuals at maximum. But when we place more than 8 or 10 individuals, the software make many interpolation and fail recognizing the position of the individuals. Mm-hmm. So 8 individuals is a optimized number of individuals because I've been checking it and I made many tests about it. So this is because I spent 1 year with the software and the building of the recording system.
But I just want to add that, yeah, so we have these kind of logistical limitations, but also we need to stress that these individuals, they are in the field and they found in small numbers like a new resource that is ready to be infested. And so these populations are relevant out there in nature, okay? So they can find a big storage of grain, and that will be like an explosion. But in other cases, they live in pine trees, small places where maybe the population sizes are like this. So these kind of small populations that are more or less interconnected through migration is the real situation. And this low numbers of individuals interacting at some point is one aspect of the relevant life of these animals. Of course, looking at more individuals will be also more interesting, but as I said, we are doing what we can at this stage, what is manageable, and from here onwards, we'll try to get bigger and to get, maybe we need to use different software that we can look at the interactions in maybe hundreds of interacting individuals, which can also happen. And yeah, so I think every picture we get from these experiments, it's useful to start building up something bigger and bigger. But yeah, no one has done anything on these questions, and so we are kind of having to work on here.
I would like to add something. Remember that the sample size of our research is the selection regime. So we have many networks associated to each selection regime. For example, we have 8 individuals interacting, and maybe we can extract some effects, and the effect maybe has a low size, okay? But we have 256 network which represent this small size of effect. So we magnify the effect by the sample size of the total of networks, not by the individuals.
Yeah, so actually Miguel is suggesting in the chat, actually something that seems to be opposite to what you just said. So he's saying, I see it's fine, but perhaps it would be better to focus on network metrics of the individuals rather than properties of the entire network. So that is an interesting point to discuss.
Yes,
certainly. Yeah, we need to get together and talk about this, because that's a certainly very nice and useful suggestion. And yeah, we need to have a chat, and of course we'll get some insights from you, Miguel, that you are an expert on this thing. So we are very happy to hear what you have to say. Yeah.
Nice. Good. So does anyone has more questions? So yeah, well, I think that's it. Next week, we won't have a seminar. We are taking a break because we were supposed to have a feria, which we don't, but we are taking a break anyhow. And then our next seminar will be on the 29th, Thursday again, and on YouTube. Paco Garcia actually will be presenting his research. So we are looking forward to have him give a talk as well. Thank you everyone again for joining us today, despite the change of a schedule. We are looking forward to see you on our next seminar. Bye.